control analysis, and second is the metabolic networking.
Yeah.
It is the first 1 hour lecture, and then after lunch time another 2 hours, and sir is the 1 hour. So what I'm thinking, after 2 and the 3, sir will cover his lecture, and 3 to 5 I will cover my MCA. At the 5 p.m. we will go CIL to see the HPTLC and the LMR. Yeah, please go ahead.
Okay. Okay. Let's start today's the session. So those two, I will combine the two. Today we have a metabolic network and also metabolic flux balance. So from the previous slides, we talked about how to identify or how to identify the metabolite. Is metabolomics kind of methodology and also different kinds of machinery we can use for the purposes. We then use the GCMS and also national LCMS, and widely using of LCMS for the metabolite. And we mentioned those sample preparation very diverse property, and of course you need to adjust your preparation method to make the sample fit or reach the best resolution in LCMS or GCMS. So all those together in previous in past 3 days, right? Now so we continue from to another level. So this level, I think, is from the machine, manipulate machine to manipulate our knowledge and manipulate our thinking and creating the new idea. So this is very, very important, actually. So everybody knows that, like I told you before. So metabolic engineering and metabolic analysis, we call it metabolomics. This analysis technology will be more and more easier, just like a photo. Photograph many years ago, we need very special professionals to take photo for everyone. Today, you need the cell phone and also previous the camera, smart camera, digital camera already makes everything very easy. You don't need to adjust the focus, the automatic. Everything becomes automatic. So you can imagine in the future, the manipulating of those LCMS, GCMS will be much more easier. Those machines eventually will be automated, will be machinized, will be manipulated in pipeline. So will be very quick. Finally, the software will also be enhanced. Currently already emerging so many artificial intellectual the application in the compound identification. So only we want to know about their methodology, their principles. We understand that, we control all things. So we understand the compound, the chemicals, chemical structures, what is their principle for the MS, for the charging, and for the unstabilization of those metabolites in the electric field with high energy and broken down unstable generated fragment, and then from fragmentation, and we can the technology of developed many different kind of ion detector, and also different kind of mass spectral ion trap, orbi trap, or other triple photo, those to increase the sensitivity and increase the capability to one time detect so many metabolites to make comparison in one sample to another. So all those technology, you don't need to worry too much about that. The thing you worry about is your knowledge. That's true. Why? Just from one of the previous, the very famous genius, mathematician, the very young person in India, scientist studied mathematics. So, yeah, he can called yeah, It's very famous. His mind. What I'm trying to focus on, trying to think that mind is important. Mind is important. That is the superpower. It should be the machine can never mimic your mind. So enhance, empower your mind is the destination or final target of this process. It's not just for learning the machine. We cannot catch up the machine. The machine just extend our capability. Like we try to move, we try to travel. So many machines, bicycle from earlier time, now the vehicle and flight, we can reach there. All those just men developed different tools to reach our goal. Yeah, from Spasher, we want to go to long distance of another place. And another, like the machine, LCMS or GCMS or others, temporary ICMS, all those Q-top or orbital, all those try to extend our vision or feeling or all those technology. We want to detect in this universal what kind of unknown compound. And those unknown compounds maybe have some function on our health. That's later. Next today we will also mention this. So eventually those compounds we try to improve our health, in the healthcare or nutrition. First nutrition, we want to survive. But we want to survive better. That's why we want to get rid of those abundant lipid or the sugar. We want to get the more health because excessive of sugar, lipid is not good. But the compound which improve of organ function, renovation of organ, live for long, that's our purpose. So that's the plant, medicine plant developed. And the LCMS eventually will use for improve the yield, improve the food, plant food nutrition, and improve the medicine, the plant, to make it human live well. So I think the power of those, in one word, is improve our knowledge and improve our vision. Yeah, using the science technology to find the new compound and eventually to engineering to fortify the nutrition function or fortify the medicinal effects function on the medicinal plant or food to serve the people, the healthcare. So that's why at this class we talk about some knowledge or we reviews earlier. I try to cover as more metabolites as possible to give the overview of any achievement we already made in metabolic network understanding and also metabolic the flux. Yeah, this metabolic flux and their balance is very important. We understand some, because the plant actually is quite magic. Many of them we don't understand. So we don't know why we try to increase one compound, but we meantime decrease the other one, which we don't want. Undesirable trait. We try to increase the yield, but then we found that we lose their resistance. Lose the resistance to the environment. That's always a kind of a trade-off. So the balance, metabolic balance also is in same meaning. When we increase this compound, another compound decrease. Particularly in the same or in parallel layout, the pathway. So that's this lecture we'll talk about. So metabolomics data revealing the metabolic flux shift, because from when you overexpress this enzyme and then you found that you decrease another the parallel the end product, parallel pathway for another end product. So all those from unrevealed from metabolomic analysis, and we also see some reprogramming under natural plant under different stress conditions. Yeah, second metabolite doesn't matter, alkaloid or terpenoid or flavonoid. You can see the shift of the metabolite by synthesis pathway, and some were repressed and some were upregulated. The redirect direction also similar pathway, and also like flux partition. You know, when we do the seeds, we see our crop getting mature, or after flowering, the seeds feeding during this seed stage. The many metabolite, the partition, flux, metabolic flux, partition that have occurred, but all those actually regulate.
So please increase the slide number.
Oh, I.
Change the slide.
Oh yeah, sorry. Okay, I will, don't touch this because this is not synchronized. So, okay, yeah, that's metabolomic data. I mean, you use those data to reveal the metabolic flux shift, reprogramming, or redirection, and also partition. Those all the nature happened, naturally happened until we use those metabolomics tools to unveil how metabolite change in comparison with untreated or treated or under different treatment stress conditions. Then we will also measure the metabolite pathways, new metabolite synthesis, and discovery of gene enzyme underlying those metabolite change. And of course, those gene and enzyme are involved in those pathway, and also in novel pathway leading to the new metabolite, or even we try to create the new compound using this plant enzyme put in this plant. I mean, this is just the currently called synthetic biology. That's for even new novel metabolite the creation and to meet our nutrition or the pharmaceutical purposes. So unknown bioactive metabolites, this is related to the nutrition or pharmaceutical and new regulatory mechanism. Those just to try to get the basic knowledge. Then eventually those any new and mechanism or new genes, those basic science will be applied, will generate the new industry of. So pretty much we should believe those since the knowledge never looks like today. Probably we cannot see any usage, but in the future you will see those knowledge like a brick block building up, eventually finding the civilization of human advance. This is the review paper I once wrote for in tea plant, because this tea plant, the reason I study tea plant from shift from other medicine plant and also soybean, just because the tea plant enriched so many different kind of metabolites. They also have alkaloid, like pure alkaloid caffeine or theobromine, and they also have supporting very enriched, not mentioning the catechins, so enriched like 8% of dry, 8 to 10 or 18% of dry mass in tea leaves. So this very heavily, so it gives rise to so many questions, interesting questions. Why those leaves enriched so much concentration of those catechins? Why the leaf still grow? They're not self-toxic. So those storage or transport is kind of also the greater interest, but not being unveiled. Why they can store it at such high concentration, right? But the leaf still grows. So how those condensed staining or catechin acetylcysteine and the flavanol, all those give, of course, we taste the teas, give different the taste. And also, shigmy pathway, through the tea plant, actually we can understand a lot of the shigmy pathway leading to the hormone, like salicylic acid biosynthesis in chloroplast, and also leading to biosynthesis of three aromatic amino acids, tryptophan, and tyrosine, and phenolphthalein. Now we know the phenolphthalein is a major precursor for phenylpropanol pathway, which is both for lignin and for flavonoid biosynthesis. But in tea, specifically those shigmy pathway is leading to the gallic acid biosynthesis. It's very extensive accumulation of gallic acid, not only in the wild tea plant, they extensively accumulate those different degree of gallowation on the sugar, on sugar backbone. The hydroxy group can link to the maximum 5 gallic acid, the ace form this ester. But one of them, the beta gallicine at the middle, the pointer is at the middle, beta gallicine is the precursor to as an acyl precursor, acyl group donor for the for the EGCG or GCG, for those compound biosynthesis. So those this very significant pathway in tea plants, EGCG is major component than EGC and ECG. All those. And then also later on I will also talk about the tryptophan and tyrosine. Tyrosine derived pigment in plants. The last actually yesterday, the day before yesterday, we talked about the betaline. The betaline is naturally another pigment that exclusively, I think exclusively accumulated in another group of plants. We call the carrarines. But anyway, they're like one of the typical plants in that family is the cactus. You see here we can see a lot of cactus. Dragon fruit also is one more fruit we eat from a cactus plant. So this is the theanine. As long as you drink tea, you are interested in this compound. And the GABA also in tea plant concentration is high, is non-protein amino acids. So this is a major target of metabolic engineering or for the high quality green tea selection or breeding from different the cultivars. So this is the caffeine or theobromine, theocrine. Recently, from some plants, some tea plants actually have a continuous methylation. From xanthine, the yellow color or light yellow color is the primary metabolite, very critical for the purine. The purine, DNA, RNA, or other very important. So those are poor in plants or in animals, all the basic primary. But the mutation or some kind of evolution generates the 7-methyl transferase. Those generate the 7-methyl xanthine. So this start step-by-step methylation and give the caffeine. So those are very common. Those steps are common in different caffeine-producing plants. Even they evolve independently. This is called the convergent evolution. Yeah, from these different precursors eventually generated. This aromaterpenoid is very complex, not only monoterpene, diterpene, and also leading to the some higher degree of C5 unit. This linkage generates carbon 30 or carbon 30 or carbon 40. Carbon 40 is the rubber, right? So in some trees have very. So in tea plant, we mainly focus on tea aroma, monoterpene, or sesquiterpene. Those volatile compounds, they contribute to the flavor. So from here, I will just use the several slides to show the alkaloid or different metabolite. What for the metabolic pathway they identified, and of course they use different tools like metabolite analysis and comparison of metabolites and biosynthesis. Those the purpose is try to understand the origin of those alkaloids, just like caffeine in tea plant. We also try to understand why some wild tea is low caffeine, I will also mention some, but most of cultivar very high caffeine. Those high caffeine actually is necessary for our as a nature the plant drinking materials like tea or cocaine or caffeine, because without the caffeine, probably not so many people are interested, just because we can stimulate, we can refresh us when we get tired, we drink this cup of it. Actually, this is major. Like catechins everywhere. Of course, in most plants, the polymer, like condensed cannons. Only in tea plant, this is very specific. Also wine and also salmon. Those plants have some soluble catechins, but caffeine is the core. So one of the pure alkaloid. So in alkaloid study recent years actually was speed up because alkaloid have very strong bioactivity. The most of them can be used for medicine. When we get the cold, we drink some medicine for reduce control our salt uncomfortable. Yes, so those are very common. Like those. Last year, the day before yesterday, we mentioned cocaine and cocaine-related metabolite. So you already know the pathway. Actually, the demonstrated pathway several key enzymes to synthesize those published in Nature Science. Those just because it's so significant to understand the small molecule have so strong bioactivity and have the pharmaceutical application. Actually many of them. This also, but they're toxic. But every compound we know is medicine. Every medicine has side effects, just like this. This actually used in many purposes. Like actually in China, this medicine is used for the person, the woman trying to give birth to a baby to enlarge the bone. To get some person have difficult. Some lady have difficult to give birth to baby. Then need to use some medicine to enlarge the bone to give very small give birth to a new baby. So this medicine, but very toxic. That's for sure. That's recently they also demonstrate the biosynthesis pathway and reconstruct the pathway in tobacco, benzyana. So this is another compound actually. Erythropoietin. It is also the compound, alkaloid, but very toxic. Just recent, less than 10 years ago, in Singapore and I think Malaysia, Thailand, they identified this compound as a major carcinogenic compound. They can cause, I think they can cause DNA nucleotide mutation from, I think from A to G. Yes, but you call the DNA mutation. They already identified. So this Chinese, so many Chinese medicine have this component. Yeah, different herbal. Many the herbal medicine have this. Finally, the China herbal medicine administration we just delivered a notice that some of medicine, traditional medicine, will withdraw from market just because this you take too much, you just get kidney, liver, or disease, or even cancer. It's very strong. Now, science paper also mentioned this specifically can induce A to G may be wrong, but it's very clear it's for sure. If you find you have mutation some gene from this specific mutation for A to G, it definitely causes this compound. Yeah, so just a single compound can cause the mutation. This just raise of caution for the medicine plant. If we don't know clearly, we can better be careful not to use the plant. I guess this is the traditional medicine need to be improved just because the Western country, yes, their principle always to medicine have to be clear components and also biologically the medicine effects should be very clear and how they metabolize in the human body, any side effect, how about the toxicity, all those evaluate. But some our crude medicine, the herbal medicine.
Belongs to class of FLux.
Yes,
They are
yes. There's a nitrogen group there.
Yeah.
But actually they're also kind of like polycation synthesis. She belongs to polycation. Yes. But I think the nitrogen group is very important for the bioactivity. Yeah, always alkaline. Just you add like Taxol. The Taxol group also we know. The Taxol in anti-cancer medicine, right? It's very famous.
Taxol and paclitaxel.
Yeah, the bioactive group, functional group actually is phenylalanine, from amino. Yeah, so that's why the alkaloid was so important. This is one paper published in Nature. It is the old time they mentioned the switching the biosynthesis, but later on they did not continue identify those. This long time ago published the paper, but those steps haven't been unveiled, and some enzymes did not identify. So here they use the genome sequence of this plant and generate a huge data and start to demonstrate which enzyme really can generate and which pathway, what is the intermediate of this enzyme reaction or biosynthesis pathway. So later on they demonstrate the pathway. This is the Nature paper in 2020. You see, those big scientists always focus on the many years ago the unknown pathway, like published in Nature 1970. But the compound is very dramatically strong. You have strong physiological activity. So it can be used for different medicine or used for toxins. That compound in agriculture used for pesticide. Yeah, for some the alpha to some insects. Yeah, need very strong, very effective. That's why, but it also toxic to human, right? So they demonstrate the pathway, identify the enzyme, and from each step, of course, they also use the metabolite profile, use L06MS or GCMS to identify the enzyme activity in each reaction. Also to confirm, because this is the compound is shown in several different plants. They also can do the plant metabolite profiling and enzyme activity assay to show indeed some plants they have lower and some another plant have higher. So you can confirm similar the gene expression level also correspondingly lower or higher. So those from naturally confirm the metabolite, the genes and the metabolite, their relationship. This is for engineering the chopping. We know chopping, right? The kind of anti-call. This also in nature communication. Recently, there's a lot of interesting results come out. All the alkaloid. So they also reconstruct the pathway and demonstrate how they can use the different biosystem like tobacco, benzodiazepine, or other plants to produce this compound. This is for biosynthesis of the tropic alkaloids. Have different added products. Basically, actually, as we know, the alkaloid, all the alkaloids are derived from amino acids. The alkaloids in plants is very, very complicated. With many different the amino acids, lysine or the tyrosine or other gives the different backbone than to continue the cyclization or methylation or other hydroxylation to generate this metabolite pathway not so complicated, but each step, but each end product, the biological effect and the pharmaceutical the value is high. So that's why so many people now work on this. Yeah, this is also the talking later pathway. So step by step, this is from after methylate the product for methyl AB and generate the tropin. And this is another paper to show those subcellular localization. They usually specifically localize in some cells. Those cells should be able to storage in specific subcellular compartment to make sure not toxic to plant cells themselves. Because it's very, if the compound is biologically effective, very strong, then should be also toxic to themselves, because our human body cell or plant cell basically have very similar the life activity, right? Yeah, this is from different plant. Also, can generate the cocaine and just like yesterday, the hygiene. And the hygiene. This is a paper. Actually, later we will also talk about the compound identification of this pharmaceutical roses. This Nature paper actually is from Owner, one of the very famous scientists previously working in Joiner Center. I think now she moved to Germany. Yeah. They also use the different, you see here, they also use the TLC you use the TLC to identify those compound. Yeah, also those TLC or GCMS, or then finally use if you have enough amount of the purified or half purified compound.
So I want to ask, how much of the time gap should be taken to analyze these subcompounds?
This depends on you. For example, you finish one time enzyme reaction or extraction. Actually, you can do the. I think later we can study.
This is very important. Actually, I want to know. Even the students want to know how much of the gap should we keep from one derivatization to the next.
Yeah, usually you can analyze at the same time. But if the step by step,
Yeah, we want to go by step.
yeah, that's depend on the work efficient. For example, first time I get the compound I try to identify, I want to use the simple method, TLC. I want to quickly develop. But TLC also have different the solvent to develop, right?
Yeah,
To extend. Then you need to use different the developer to develop the color to identify the compound. But also you can use the UV detector, put on the UV. You can manipulate to show the figure. Then you can cut the spots, then scratch out the silica, the powder. Yeah, then dissolve in the hydrochloride or destroy in methanol. Then do further analysis. Just one simple HPLC, you can find the compound with different the picture. This is correct. Then if the compound is interesting or you extract the compound, and you can do the bioactivity assay. If you have some model you want to interested in what kind of model to test the biological activity. Then next you can actually the single point definitely a mixture, still a mixture. So you can log down the LCMS or GCMS to identify what those compounds are. So the if this can be used for GCMS, depend on your developer. When you use the kind of hydrophobic, the developer like SEO or chlorophyll, then you mainly develop some isolate or separate the compound is the oil salt, right? So, but if you use the methanol, probably those develop the total or other. Yeah, those also can help you to identify get preliminary information about the compound, the chemical property. So all of those actually can. I think this one you just try to find the synthesis I put in substrate. Then if I get the target compound, okay, that means the activity then use GCMS to confirm. Yeah, then if my enzyme activity very high, I can put more substrate, finally accumulate enough, then log down the NMR. Finally, NMR is very important for the structure, chemical structure identification. So those you can get the structure, right? Because substrate is clear. You know this, so very easy to identify. So this is biological for the LCMS analyze those the compound benzoate. We know the Casarasa rosas is a very famous anti-cancer compound like wing blasting or wing twisting. So windoling is one precursor for generation. The G10H is one of very important enzyme. The whole pathway actually is clear. I think it's clear. Actually, this professor is involved. This actually pathway is very earlier. pathway of generate the precursor. Yes. So they demonstrated, then they think this is a very important discovery and submit to the nature. This again to find the mechanism of the chemical enzyme reaction, how they catalyze what's the intermediate to find out. This is also the paper to demonstrate. No. This actually they use another the fungi reductase to generate the new compound, artificial compound, like belong to like things with biology. Yeah. Synthetic biology. So generate new compound. Yeah, that's really exciting. That means we use different. Like here, use the fungi. Because each enzyme from this plant, you definitely generate coupled with other enzyme generate this compound. However, if you change it to other enzyme, most likely with similar result. However, if you continue change, maybe something happens. So they use one enzyme reductase and generates a new compound. That's exciting. That's I think one of the hot point which are attributed by to the nature editor to get published. The carotenoid also I will just quickly go through. That's a very common pigment in plants. The several carotenoids, flavonoids as pigments, and also the betalain. Later on we'll also mention those. All those are actually very interesting. You will understand how the flavonoid, one of the major group of flavonoid as cyanins, how compete with the betalain, why they exclude each other. In plant, you cannot have both as cyanins and the betalain together. They always show that only have a flavonoid. This most plant have as cyanins, but only in a few or specific group family of plants have a betalain. Yeah, this is one publication. Showed some years ago, 19, just before COVID-19. So they published a paper identify R2R3 mutant factor interact with TT8 and WD40, TGT1 to control the carotenoid biosynthesis. They use first they use the mutant to find that some this NIB transferrin factor, the defects of the mutation cause the flower color change. They did a metabolic profiling, found flavonoid change also, but the major change is carotenoid because the lutein they decrease dramatically. Here you can see the compound, right? Lutein decreased. In the bottom, the K panel, they set the lutein episode and also Willisexin. This also the carotenoids, the pigment for the photobiosynthesis. All those decrease dramatically in both the independent mutant line. That suggests that carotenoids were dramatically reduced. They are associated with this new transferrin factor. Then we found that the MIB transferrin factor belongs to R2, R3, very closely related to the LAP1 or PEP1. Those transferrin factor regulate the S-cyanine biosynthesis. Then they also confirmed they can interact together to form a MBW complex. Finally, they also use those activity assays suggest the WT1 and TT8 interaction with TTG1 to form a ternary complex and regulate the carotenoid biosynthesis pathway like PDS, VDS, and other genes like CYP73C is one of the major enzyme for synthesis of the protein. So this is the pathway, and suggest those enzymes there. I use the red arrow to highlight the deuterium pathway, which is shown in this metacardial contracture flower, and they also test the enzyme. The enzyme coding gene, the expression level indeed dramatically reduced in those mutant corresponding to WT1 mutation. But the overexpression, you can see the EOX overexpression can cause the upregulation. So the downregulation of biosynthesis enzyme is correlated to the mutation of the regulator. And the overexpression and also correlated with the enzyme gene increase. So they use the reporter assay and to express the WT1 in bacteria, then put in a vector, and together with the reporter gene, the promoter drive the reporter gene and luciferase transferase of GMP. You can put together, like infiltrating flacle cell. Then you will see the transferrin factor will bind to the promoter region and gets the reporter upregulated. When you spray substrate of luciferase, you know, the firefly, they will show the luciferase signal dramatically increase. That means the transferrin factor binds to the promoter, and the promoter is isolated from those genes, the biosynthetic genes, so they can identify which by metabolic biosynthesis genes have this promoter can be bind to WT1. So you use this promoter transcriptional activation assay to confirm the regulatory relationship between the transferrin factor and the target gene. So this is both mutant and also complementation and also I think overexpression. And they also did the interaction assay, confirmed that the only WT1 work less very active. However, if you combine the TT8 together, put the 3 transferrin factor WT1 expressed in a vector, and also TT8 in another vector, and also then the TTG1 in different vector, then probably combine the 2 or 3 the aerobic bacteria individually or in different combination together in infiltration with the reporter gene, then you will see this put together have maximum effect. That means indeed the protein interaction have the effect from a complex to regulate the biosynthesis of keratin. Here the lutein. This is this is another transferrin factor that regulates another flavonoid. It's the RCP1. In different plants, there are different RC3 transferrin factors because carotenoid is always present in the plant, right? Together with the photosynthesis, because this, usually we think the carotenoid is the kind of protective of organic photosynthesis, the organ, like chloroplast, to make sure the protein or the light harvesting complex or other complex the proper function. But yeah, this is similar. Basically, people believe that. So carotenoids is very widely. This flower also mutation one of transcription factor R2, R3, NIPA, RCP1 that got reduced carotenoids. They use similar way to confirm some of downstream genes were targeted. But however, they also confirm that overexpression of this gene, and also can activate some assigning biosynthesis gene like NRS, PFR, or NS. So that means that this RCP1 actually regulates both the carotenoid and assigning. Actually, this paper also showed that have weighting effect. I mean, at the beginning we showed the mutant flower. The mutant plant, the flower very like a poor white or the green white contain those carotenoid, but still there's some assigning also. The flavonoid also lows. That means the mainly regulates the carotenoid, but also regulates assigning. Yeah, this. So those both paper actually confirm this point. That means probably in most plant, the regulatory mechanism function in similar way. I think this one, this our publication, we also show in tea plants, the leaf also has a lot of carotenoids. So carotenoids were also regulated by one of MIB transcription factor, we call MIB 110. Yeah, this is just a phylogenetic characterization of how MIB transcription factor we characterize this transcription factor regulates the carotenoids in tea plants, in leaf. And there are also the partner TT8, the medical camellia, the synesis, the TT8 homolog, and also have interaction with the MIB transcription factor, the MIB 110, to regulate carotenoid biosynthesis. The phylogenetic tree also shows that they actually class together in same homolog. That means the protein sequence similarity basically define those MIB transcription factor have similar function also in TCAM. The flavonoid is the. Did I go too fast or slide?
Okay.
Huh?
Okay.
If you go too fast, then just tell me probably we quickly pass through the alkaloid and the carotenoid and the alkaloid. Alkaloid, of course, that's very common. This is signing. They are also much more diverse than than carotenoid. Carotenoid is not so many different kind of in different plants. They have different dominant molecular species accumulate. For example, in tomato, they probably use red color stuff. What in tomato? What's the major carotenoid? Yeah, lycopene, right? However, in other different colors, maybe Luteonia is the most common to the other plants. Yeah, so in some also, in some plant is cyanidin derivative, and in some the purple plant, purple flowers, maybe delphinidin. They show the purple, but in some red or light red, probably cyanidin. Those However, the methylation or hydroxylation, the different hydroxylation, mainly the beryllium hydroxy group number, mainly different, like the delphinidine have 3 hydroxy group, and the cyanide have 2, the pelargonidine only have 1. You can see here. So those define the different the compound of the methylation. They will show the different color on the same, under the same pH. The pH can affect the coloration of this PPT actually I showed us before. When we work on the legume plant, mostly we have this kind of pathway blending together. Those pathway have if you have more pathway in parallel, of course you have more the computation, metabolite computation, because the precursor you have only very limited. But in different organs, like in flower, in soybean flower, there's some the purple or some the red, yeah. There's some even white. So because soybean is one of the major crops, so people actually gather with the seed color, they define some the QT of the molecule to find some the genetic loci to determine the seed color or flower color. But they also found, I think later I will also show that some color, some acetylenics, the high acetylene plant usually have a lower acetylflavor. Yeah, so this is just because the branch to acetylflavor, here is the brown color, and the acetylene, the downturn, the purple over, we have composition on the metabolic precursor. Metabolic precursor limited. You either go this way as a major product or otherwise go this way. But if both are very strong, then probably still have one of the dominant. But if we see some acetylene seeds very high acetylene, then acetylene will be less. They always, because the plant, there are other strategies to control the metabolite precursor. We don't go too many from phenylalanine, spend so much energy to produce those. They only can control the certain flux to go either acetylene or acetylene. So otherwise, the metabolism will be primary, right? At least secondary, until you have function for defense. Otherwise, there's no method to produce the metabolite because the energy and substrate costly. So this we confirmed when we use medical transgenic the hairy root to generate one of the genes, the empty part. This gene actually regulates the acetylene, can activate acetylene biosynthesis. Mainly also can activate here, activate both this side, acetylene ANR, to both acetylene and epicatechin. So the catechin or epicatechin actually to both products, their major, the precursor is acetylidine. The different type of acetylidine have cyanidine or pelargonidine or delphinidine, this in blue part. So the blue part actually have a competition with the brown acetylflavor. So we confirmed this. I know actually beginning we don't have too much information about the legume plant. Also, there are so many different types of flavonoids. How those flavonoid and products will compete with each other, because we know, just like in a family, the everyday input is the same, is balanced, stable. Yeah, until there's some very strong light or some, the plant adjusts probably since it's more acidic, since it's more flavonoid to absorb the UV light to reduce the damage of the UV light on plant. Otherwise, they always very stable. Like metabolic flux don't go for too much accumulation. Otherwise, it will affect the plant growth, because the actual energy or substance go to the secondary metabolite. Secondary metabolite, this over accumulation will definitely cause plant drop. When we always flash one over like PEP 1 control that, as a result is here S-cynin, as plant become drop. And purple, yes, true, plant is very purple. It's very dark, but however the plant is drop, it cannot grow. That means the energy the plant uses to synthesize carbon or other amino acids like phenolphthalein. If you go to the flavonoid too much, then you have less energy to produce the cell division to grow up, right? Yeah, that's also for human, it's true. If you have nutritious, you have too much, lot of money, then your family grow very well, right? They grow taller or healthier, but however, in poverty, then the growth, the nutrition deficiency show up. Well, not. So in the plant also need a competition. We have like competition. So that's how the metabolic flux, the balance needed. The balance not only in the regular way, under normal condition, this flavonoid by synthesis several burns compete each other. We also will show that S-cynin compete with the catechins in tea plants, obvious. You can see that if purple leaves, you really have less catechins. Yeah, this is also obvious. But in soybean or other legume plants, if you have more S-cynin, then you have less isoflavone. If you have much more isoflavone in the root, then you almost don't have no S-cynin. Yeah. So that's in normal condition. But under stress, the condition, the plant need to adjust the defense and growth to give. So in this way, we show that antipart.
We have to go for the lunch.
Oh, okay.
And you can continue one hour. Then you take the rest.
Okay. Yeah, okay. This shows the competition. For example, we always test this thing. We see the S-cynin increase and the catechin increase. However, in another study, we can see the isoflavone, the white bar, the dramatic decrease. Those lines are independent lines, but you see some of highly expressed, or transgene highly expressed, some of transgenes lower expressed. The plant, you transgenic plant, not always high. They're not even. Some of them, I don't know, probably when we do transgenic, your vector insert different position of whole genome. So have different expression level. So the higher expression level causes your target compound increase. But in the meantime, this target compound accumulation will affect isoflavone. So when we get this pattern, like this, you higher isoflavone, you lower your target gene expression, like control. And correspondingly, the target enzyme shows a similar pattern. Oh. Okay, later on, we will, okay, yeah. Okay. We'll quickly go. Thank you very much. I think
which are the paper which are really good in the second. Someone is staff we have Professor Joe. One is the faculty in the JMU, Professor Ashish Gandhi. He also did the postdoc with the sir in the USA. And then second is the BU University, there is a Dr. Misra. He is also the professor. He worked as a postdoc with Professor Joe in the US. So yesterday we talked to them and they told me if you are the appropriate person who is inviting someone who is doing the work on the metabolomics. So they both told me this thing and yes, whatever the tips knowledge you are taking from here regarding the metabolomics, it will be definitely useful for all of you in the near future. I am just trying to give some theoretical knowledge also, but sir is frankly giving you all the practical knowledge. He is explaining what type of the final data you need in your manuscript and what from where you get the idea to do the work actually. So what is the paper he is representing? If you are in the early stage of your PhD, so you have to find out where you have to work, what type of work you have to do. So there is a break for 45 minutes around and 45 to 50 minutes for the lunch. Then we will come here. Sir will continue 1 hour and the rest of the time I will take care of this MC and other things for you. So thank you for this lecture. I think specific for you can say metabolomics they are using for the traditional medicine. In India we are using the Ayurvedic, herbal, Unani and a few Chinese medicines are also use the secondary metabolites. So sir will explain that what are the metabolites which are used and how they are useful for the different medicine purposes. So please go ahead, Professor Jay.
Yeah, yes, sir. Okay, I will just a little bit change it. Yeah, because previous, as I said, we still try to understand how to identify the particular compound through the LCMS. So previously, I also promise you that I will show you using flavonoid as an example to see how we digest, how we analyze the iron peak. We can predict the molecule to identify. Usually identify the compound is very important. Also in medicine, traditional medicine plants, in the herbal, the herbal medicine. So it's very important. However, I think I will just shift that part to tomorrow. Talk both identification of the components and also in traditional metabolite, how in traditional medicine, how we focus on the identify the metabolite in complex. For example, in herbal, the medicine usually is not one plant. They have other different plants from specific organs, probably root or leaves, some seeds, or depend on. So those the very different issue together with how we identify those compounds, how we accurately measure those compounds. Use the LC-MS, HPLC to do the quality check or do the functional components analysis to make sure. So those are all is one of the major steps in medicine plant pipeline or in traditional medicine. Some department they want to control the property. You make the traditional medicine use this component. You said use this 4 or 3 leaf or some the plant organ to mix together. Use the ratio was assigned by the medicine plant or guided the medicine. You said do that. You do that. However, the products, those products really contain the major component or bioactive components contribute to the clinical use or clinical effect of this. So the department want to make the quality control, and the company itself want to also need a control to those property, the quality. So those I think probably need tomorrow. I will talk about it, and I will I will continue demonstrate so the whole metabolite, different types of metabolite in the plant, so that we can have a basic idea. Those compounds, usually the synthesis or other the next step we focus on the medicine, traditional medicine or medicinal the plant, and also compound identification, quality check, and the clinical. There are probably several examples how people identify in this very traditional thousands of years ago. People developed this medicine and use different type of medicinal plant component to find their effect. However, in modern time, use high technology, we need to confirm which components really affect them. Which components, or which 2 or 3 components together give the clinical effect, can treatment disease or cure any the symptom to recover the patient from the disease sickness. So those actually I want to combine together. And this afternoon I will just use 1 hour because after Dr. Wayne will give it. So we will quickly continue just to give you a brief idea or principle of implant, also how those metabolites change each other. So then you have a deep understanding of the plant metabolite. Then you will focus on the usage in the traditional medicine plant. So yeah, this morning we talked about that. So we just use metabolic engineer, always express the transferrin factor, or we always express a typical enzyme. You can see the components change just because the metabolite, the pathway in parallel or in different, they will compete, they will interact with each other because the total precursor, metabolic precursor amounts are limited for plant under common conditions. They cannot use too much, otherwise the plant growth will be affected as a plant in whole. So they have strategy to change them, to manipulate those. This is another example that we identified empty part from the mutant seed phenotype. And we see the mutant seed color difference. Then further we identify the compound by identify the function of this gene. When we always express the anti-par gene in metacardial truncation hairy root, not only in seeds, mutation, deletion of this gene causes the seeds color change, but also when you use other strategy, for example, overexpression in transgenic alpha heavy root, you can see the metabolite change in gas control only those compounds. However, when you overexpress anti-par, the other compounds come out, like epicatine glucose modification, and also quercetin and the camphopheric glucoside dramatically increase. That means just one metabolite, if you the one transferrin factor, as long as they expressed in the organ, in the tissue, they will activate the reorganize the transcriptome. And somehow in genome, they activate those metabolic gene and change the metabolite profiling. So you understand why under some circumstances, like stressing or job stress or other disease, they suddenly change the metabolite profiling. Just because there's some transferrin factor to control the metabolite was changed in their transcripts level, or will be upregulated or will be suppressed. So that's how we understand why under different stress condition, the metabolite will shift or reprogram just because transcription factor will change. So this example just explains how the transferrin factor is very important, like this need regulates the flavonoid, and also the previous this morning talk we showed another leaf transferrin factor regulates the carotenoid biosynthesis, and in the meantime slightly regulated as a signing component. So this will still use those hairy root in soybean hairy root. And we showed when we overexpress the anti-par, we found a decrease as flavor. Because in soybean, they don't generate the S-signin. The hairy root, they don't have color. They just show the white, but they usually accumulate a lot of isoflavone. You saw it in heavy root. However, when we overexpress this C, we found that isoflavone will change. We did not added see the acetylene production, probably because the precursor you lack you only overexpress NAS, it doesn't enough. It's not enough for acetylene production. They need other components. However, we see the isoflavone repressing. So overall, we confirm those. When we overexpress the anti-PAR, the acetylene increase and epicatechin increase. However, isoflavone was repressed. So this means this transferrin factor specifically upregulates the acetylene and the pyrocyanidium epicatechin pathway, but repress the isoflavone pathway. The target gene, like IFS, IFF1, 2, and also CHR, is specific for isoflavone biosynthesis. Several key gene expression change targeted by MD-PAR can change other pathway. So that's what we explained. We also overexpress MD-PAR together with LEP, which overexpress acetylene. Then we can see we increase the catechin level. So all of those, this will generate the transgenic alfalfa, which produce pyrocyanidium and enhance the pyrocyanidium production in alfalfa. Normally they don't produce catechins, pyrocyanidium. However, when we overexpress MT-PAR, and they produce catechins or epicatechin, and those compounds have a lot of hydroxy group and prevent the lethal passage blood. The stop or slow down the protein digestion in first the stomach of cattle or sheep. Those animals have two stomach. The first one, you just quickly digest. However, like this, alfalfa usually causes lethal passage blood because the protein very easily degraded. However, if you have so many hydroxy, like even check here, from tea plant, if you mix it with alfalfa, people reduce the passage blood. This is in other places people usually use to prevent lethal, to prevent the animal death by the lethal passage blood. So we use the transgenic, transgenic approach to metabolize, to do the metabolic engineering for the pyrocyanidium biosynthesis. This actually already have transgenic plants in the market, which licensed to several companies like Japan, New Zealand, Argentina have production. So this is the TT8 again, another one control the acetylene and pyrocyanidium biosynthesis. You just do functional characterization, use mutant. You can see the color, see the color and also flower change. So this MT actually previous Livonian paper, the same genes. We see the color change. Also, this MT actually involved in the keratin biosynthesis pathway because the WP1 interact with the MT. So our paper just demonstrate that MT involved in the acetylene and the pyrocyanidium biosynthesis. But another paper later, this is 16. So they later published paper show that MT1, another R2, R3 new transcription factor actually interact with the MT to regulate the carotenoids. So this different paper, in different time contribute to the acetylene regulation and the keratin regulation. So this not spend too much time, but anyway, in a similar way we demonstrate this is for the to demonstrate the computation in downstream. In another upstream one, there are another computations between the two branching pathway, like share the same precursors, like for example here, from flavonoids. The both dihydroflavonoids, three different types, myrcene and the quercetin and the camphophylline. The dihydrocamphophylline, dihydroquercetin, and myrcene generates different flavonoids. Flavonoid catalyzed by the FLX. However, this pathway downstream go to acetylcysteine and chlorosine. So they catalyzed by DFR. In this point, branch point, the precursor is only dihydroflavonoids. Then used by different enzymes. You can see the color change, color and signing change depends on FLS. If FLS overexpressed, then color become white because acetylcysteine was blocked. Because the overexpression FLS will use all the dihydroflavonoid precursor for this, for the flavonoid biosynthesis. Flavonoid is colorless. However, if you block FLS, then DFR will use this metabolite precursors to generate acetylcysteine. Then color become red and purple. So this is very widely. So we published in 2013. Now already like 130 something the citation. So actually, very much later, a lot of other plants they confirm like this. This is the cherry. In cherry, the fruit color also show like this dihydroquercetin, the dihydroquercetin to camphor over this dihydrocamphophylline to camphor, dihydroquercetin to quercetin, and also cyanide. This also depends on the enzyme what you add. So the same thing is pretty true for other plants. That's why so many people, scientists confirm in their plant also the same principle of computation between the flavonoid biosynthesis and the acetylcysteine biosynthesis are competitive at the FLS and the DFR enzyme because they share the same precursor. So I will just use this quickly go through. This is the T trichome. So we just want to show that the trichome is one of the most important traits for tea plant. If the trichome is more, the tea shoot or young leaves covered by more trichome, they will have a more stronger flavor, and also the inside the bioactive compound and more. So the flavor of both the smell, aroma, and the taste are better. So the trichome, that's why we call the trichome is one of the most important traits for tea plant. So later on, most those modern tea cultivars are covered by thick and highly and densely spaced trichome. So those give the actual flavor. So we found that actually trichome contains not only those ECG, but also contains flavonoid, caffeine, theanine, and other the volatile compounds. So both give the taste and the aroma. Together we call it flavor. Yeah, taste and so this trichome is very important. We also demonstrate the trichome, the density of the length are affected by the genetic background. So different plants, different cultivar have different density of those the trichome. And also it determines they have this is fresh. The previous slide showed this dry tea with more trichome or less trichome. So we also use the scanning electroscope to show that you see that photo shows the stomach and also trichome in the ectodermal cells developed. So they have a component all of the major metabolites as a characteristic to the tea plant and volatile in the fresh. So how we isolate the fresh trichome? If you study your plant, you want to isolate the trichome. How do you do that? I'll tell you a tip. We just freeze the young leaf and also stem, first apical bud. Then freeze and put the liquid nitrogen. Then we quickly use the season or use the bar, the glass bar, to scratch. Then remove the trichome. Then you will get enough. You accumulate the trichome. Those should be fresh. Then you can use it for do the metabolite profiling and use it for the transcriptome because it's fresh. They have gene. It's single cell. Those for some trichome is glandular, secretory glandular. Those trichome. So later I will show you. In some plants they have 3 different, like a tomato. They have 5 different types of trichome. Some are glandular. We can see, we can smell the tomato plant is different, but some of them are just protective, very sharp, to protect them from insects also. So the metabolite and transcriptomic also support the trichome function as a defensive, defensive organ, or will have some other function like the densely spaced trichome can reflect very strong UV light or other highlight from the damage of the young tissue. That's why they have a very strong trichome. Plus, you also give the very strong and extra flavor. That's why in modern tea cultivar, we mostly see the very high densely spaced trichome. So because of human selection and also natural selection, because tea plants, from forest, originally from forest, then expand their territory to the other place. Need very strong strategy to adaptation to the new environment. So that's why we think the trichome is very important. Later on, I think in next few days, the international meeting, I will also talk about the molecular mechanism, how trichome is developed and selected in tea plant, only in model tea plant, because their one of the key genes control trichome development, actually have the promoter, the mutation. Yeah, all the modern tea cultivar, they have one type of promoter. However, in wild type tea plant, you will see different mutation cause the repression and affect the trichome development. Actually, those really one of the mechanism underlying the trichome, underlying the tea domestication. Yeah, means human selection and also for the better resistance against the insects or the herbivores. So this we found the low caffeine tea. They have less trichome, the wild type tea. But anyway, they just have a very low caffeine because in society, right now in this world, both caffeine and tea, more and more people expect low caffeine because caffeine really makes them in trouble because they couldn't fall asleep. To modern people, probably in big city, Mumbai or Delhi, you will see many people with high income or high end. The problem is that they have good money. You thought they are very happy. No, they're not happy because they're very busy and they cannot fall asleep. Yeah, because of anxious pressure or what other psychological effect cause the sleep is very, very good. Very, very critical thing for their health. So that's why low caffeine, decaffeinated tea or coffee become increasingly popular. So we also try to pursue low caffeine tea. from a German plasma screen, I find this. But anyway, this is those low caffeine tea leaves make also very good tea, and from their flavor, use electro nose to smell those products, very good. So we just confirm the amino acid. Okay, I continue. So in theanine, in tea plant, theanine is very important. But in other plants, amino acids, high protein is very important. As I before said, lysine is very important for the corn. Because corn, the lysine is very low. So this makes nutrition. Lysine is one of necessary amino acids for the human diet. So corn, lot of parts actually have for the human food. So they try to manipulate lysine in the corn and also enhance the cysteine. Those amino acids contain the sulfur in the forage. So also in tea plants, we try to increase the theanine because this can have the emotional flux and also neuroprotection and enhance the sleep. There's some theanine pill in the pharmaceutical company or in the store, nutraceutical store. They don't need a prescription. You can directly buy those. You take some, they will help you sleep. So sleep is one of the problem for modern young people or high-end people with but we don't know the theanine biosynthesis. Initially we thought it would grow the many synthesis in root and accumulate, then transport to the top leaf, the young leaves, epigobard. Then we pick up epigobard our young leaves to make into tea. So this theanine content is about 2% to 4%. So one of the major amino acids we took from tea. We benefit from the tea because recently there are a lot of studies see that theanine have so many the benefits to human. However, we don't know this biosynthesis or the limitation. We try to improve the yield content of the tea variety. Try to increase the theanine in some tea variety. However, we already get filled because we really don't understand the biosynthesis. So we just digest those plants into different parts, then do the amino acid, all the amino acids. Use Suzuki, use one of amino acid analyzer. We can analyze those metabolites. So this is one way to analyze all the free amino acids. So you can see the amino acids actually under the 5 molar ammonia feeding or control. All have higher or lower differential distribution. So we continue check if theanine is really synthesized in root. Indeed, you can see the glutamine, glutamate, and theanine. The content are very different. The theanine level is very low, very high. The major amino acid, free amino acid in tea root, and also their content increase upon the ammonium feeding. So we think this pathway is very can fit this. And we also actually analyze the gene expression. Indeed, before the ammonium feeding and after ammonium feeding, we see the gene expression consistently reflect the metabolic pathway, which some already know, but some of them we don't know yet, like which one really synthesizes theanine. And also, we also profile the other amino acids, which are like neuron, like citrate, and oleucine, and also arginine. And those metabolites, actually we know is very closely related to theanine. But before, people don't think they have a very close relationship. Now we at least we propose, they have very tight connection with the theanine accumulation, because if you add the more ammonium, the glutamate will increase. However, glutamate as a substrate to fixation the nitrogen, ammonium glutamate use GS generate glutamine. However, glutamine is a signal molecule. In cytosol, if the glutamine increases dramatically, then arginine biosynthesis will activate because there are glutamine binding protein called P2 protein. They will bind to the glutamate and conformation change will active GAPK. Active the NATK. One of the NATK, very important rate limiting kinase enzyme. This enzyme activation means the nitrogen flux will quickly flow to the arginine. Arginine then synthesizes a lot of polyamine. So this causes the nitrogen flux to the polyamine rather than theanine. Only a small portion of the theanine increase. So this mechanism is actually very important. Now we try to understand what's the limits, what's the key factor affect the theanine increase. So if we've broken that part, probably theanine can increase a lot. So we try to understand that's a limit. That's why one of the reasons we try to dissect those pathways and also understand the different part of the root, the lateral root or root hair of those major root, the function, the components, dissection, those will facilitate our understanding theanine biosynthesis, this major bioactive components in tea leaves. So we also did the xylem, phloem, the components, the profiling, and we found that arginine is one of the major transporter amino acid transport form of the nitrogen. So makes the root nitrogen after fixation quickly transport to the leaves. So we confirmed the nitrogen, the arginine, and also glutamate, and theanine. Theanine also can transport through xylem to the top. And from the dissection of the tea leaves in different parts, like the leaf wing, and also leaf, and also different leaf position, we also understand those, also stem. So we understand after the transport from bottom, from root to the top, in the tandem shoot, and to the leaf, what happened in terms of amino acid metabolic or recycling and all those other. So, and finally, we confirm GS is very important. We use in-situ hybridization, confirm the cell, the transcripts of the GS1A is correlated with the theanine biosynthesis, probably during the transport, because if we think of the theanine, mainly synthesis in root. However, not so many theanine transport from root. Many of them actually synthesized in the young leaves. Because the major transporter form is the arginine. In the leaf of arginine, the arginine probably degraded because the arginine have only 4 carbon, but 5 carbon, but have 4 nitrogen. Yeah, means this form is very efficient transport of nitrogen from root to the shoot tip. So this efficient transport to the shoot then start to degradation, then generate urine and other amino recycling into the glutamate or amino acid. So totally we propose this the theanine, glutamate, glutamine, the transport then through different metabolite cycle, finally generated another more amino acid. So this assigning and with weighting, that means this is very interesting. Why? Because as I mentioned before, in our nature, the plant kingdom, no one plant have both assigning or both or weighting. They're mainly because the metabolic pathway competitive. Okay, the balance also have balance metabolite. So this is the vitamin, and this is the vitamic acid, the precursor, and this is the assigning step. The assigning, we know we have ACB rings, and the R group, side group can change a lot from different metabolite, different type of assigning. So this is the plant contains the vitamins. So just one sample paper recently said the mechanism underlying is naturally exclusive. They exclude each other. Exclusively accumulates only in one plant. The distribution of vitamin and assigning pigments have long fascinating evolutionary biology. So the vitamin class of yellow and tyrosine derived the pigments found in plant of Caryophyllus. Yeah, only in Caryophyllus, mainly. Also in the high order of fungi, very few, but still have. And then including this Caryophyllus family, including beets. You know, there are sugar beet and also have a red beet. Right? So people compare with the sugar beet and the red beet to distinguish. Some accumulate a lot of the sugar, but some are very rich in the vitamin. So they can make a comparison to confirm that. So later we will talk about the cocktail and also the dragon fruit and also the carnivorous plant. One of the pictures showed it. Yeah. So the major things they found one paper found the ADH mutation in red beets, showing the W7350, the amino acids from W to B. This mutation only happens in red beets, not in the sugar beets. And this mutation contributes to different property of the ADH alpha, because ADH alpha is very important for tyrosine tyrosine biosynthesis. This mutation, the paper showed that this mutation causes this ADH or PDH very less sensitive to tyrosine. Usually in all plants, tyrosine is excessive tyrosine will inhibit ADH. They don't allow too many tyrosine with biosynthesis will accumulate. However, in the red beet, tyrosine accumulation level is drastically high. So this ADH enzyme was not sensitive to, not like other plant ADH alpha. So they think this is a very important issue. At least in this paper, the title is the relaxation of tyrosine pathway regulation underlying the evolution of vitamin pigmentation in Caryophyllus, the plant. So, but this title also tell you about the computation from phenylalanine. So one pathway from Shigmid pathway, you know, the prephenate generates the agronate. The agronate is very important, but this compound also generates the salicylic acid. So one side they go to phenylalanine, another side go to tyrosine. In assigning produced plant, phenylalanine accumulation is very high. However, in that plant, tyrosine accumulation is very low, very limited. As long as you have more tyrosine was not used for protein biosynthesis, the over-accumulation quickly feedback inhibit this ADH to slow down or to stop tyrosine biosynthesis because you over-accumulate. In plant, always like this. the feedback metabolites products inhibition is very important, just like previous we said the same glutamine. Glutamine inhibits the GS, but then promote the arginine biosynthesis. So you over accumulation of glutamine just promote arginine biosynthesis, but inhibit GS because the cell tell the metabolite of some machinery. Okay, our glutamine is extensive. We don't need synthesis so much. So nitrogen fixation temporarily stop. Our they just GS but promote excess of glutamine into the arginine to store the amino into the polyamine. Yeah, this is a way to stop energy, stop nitrogen nutrition. It's very important feedback. It's almost everywhere. So this is another way you understand the metabolite, the flux, and feedback of some the computation. So they showed, you see this experiment, they showed the tyrosine concentration over the increase of tyrosine concentration only the two plant ADH alpha. One is from red beans, the BVADH is brown, and also another is light red, is SOADH. SOADH is spinach. Spinach plant, red spinach plant also accumulates the betaline. All those accumulates the betaline, the plant accumulates the betaline, their ADH alpha was not sensitive to their activity, was not inhibited by the tyrosine concentration even higher, you still have very strong activity. So that's a proof that tyrosine activity, the feedback inhibition by the tyrosine, our ADH activity feedback by tyrosine was low, was not effect in the betaline accumulation path. So this gives their very strong support. This also compared with the other, the betaline, more betaline accumulation plant from I think some of the cultivated plant. So ours shows less sensitive to betaline. So this idea is very important. There another, they also analyze the different plant, analyze the ADH, the tyrosine content. They found that all the betaline producing plant have much higher, look at this side, have much higher tyrosine. So this is the reason why betaline will accumulate in those plants. But as a semi-accumulating plant, very few tyrosine, because feedback inhibition causes the tyrosine cannot accumulate more. The long tyrosine accumulates, they quickly lead to the protein biosynthesis. They don't accumulate more, otherwise the feedback inhibits to let more phenylalanine produce. Phenylalanine production and lead to the S-cynin accumulation. So this is very different. Cause the distinguish our exclusive accumulation of S-cynin or beta. So this is another experiment show that downstream several enzyme like CYP, this P450 protein, to P450 protein D1, D6, and also DOBA, because the precursor is DOBA. Those they do the engineering for it they express in tobacco leaves. Yeah, you can see that. This is in yeast. When they co-express this enzyme, they accumulate the betaline in yeast. Because DOBA is the precursor normally produced in yeast also. So this is the overexpressing tobacco. You can see the colors or something show up accumulation of betaline. So the major components, this is transgenic plant. I think they show that those 2P450 and also DOBA. The glycosyl transferase is also important. They correlate with protein accumulation. The mutation of one of the virus-induced gene silence, the PTRV2 is the virus-induced gene silence, the vector, effect vector. When you put those enzymes into and downregulate, repress those enzymes, you will see no or less protein accumulation. So this actually demonstrates the downstream to critical enzyme. They also did a transgenic plant overexpress those three genes that cause accumulation of the protein in tobacco. That means you can remetabolize, regenerate or reconstruct the pathway in another plant. The DOBA is not the accumulation in our plant. So, but later on, the modification like DOBA, those DOBA 1, DODA1, and 2 enzyme, glycosyl transferase and P450, very important. They also confirmed that 2P450, D1, D2 is important, especially D1 is very important. Later on, they basically confirmed that, like in left side, the 3 enzymes, how the important ADH alpha, the very uniquely expressed in protein accumulation plant alpha, is very important. This mutation, they aligned up the sequence, protein sequence, the mutation W375 amino acids, mutation into B. From W to B, the amino acid, the mutation is very important. It contributes to the earlier pathway distinguish the plant will accumulate the tyrosine or accumulates the phenylalanine. This definitely, decisively decide this plant will accumulate the protein or accumulate There are also downstream to P450 enzyme like this. It's very important. And later on, the DOBA is in different plant lineages shown. So the one of the enzyme, environmental transferrin factor NIB1 is also R2R3 enzyme. So not like assigning regulates regulation by R2R3 NIB together with TG2 and also WT40 formula complex. This is very common in plant. So for the protein biosynthesis, no, they don't need TT8, they don't need TTG1, they only need NIB1, that's enough. So why this is the case? So people still, but already I think it's couple tell you from 10 years, the extensive nature genetics, several nature genetics demonstrated recently 16, 17, 18 from different aspects demonstrated from tyrosine Cordoba, then to other modification P450 or other glycosyl transferase finally generate the protein or the yellow color, the beta zesin. Yeah, those because the protein have two different. One is the protein, the glycoside, the dark red, but the yellow one also. That's why we can see some the cactus, the flower is yellowish. That's because the yellowish also not carotenoid, not carotenoid. It's just also the beta set. So from this aspect, you can understand. Okay, the competition not only in everywhere, right? From assigning to protein biosynthesis. So left side is the very common, universal assigning or proassigning in regulatory. The TT2, TT8, TTG1 to regulate. However, in this assigning in protein biosynthesis, they don't have complex. Yeah, you just step by step the limitation of it. I will quickly go through the morning one. Students actually asked about the QTL and metabolite QTL, metabolite G1. So actually, that's very important for large scale for discover some novel or key genes or genetic loci, or you find out the useful metabolite to do the like crop nutrition enhancement or medicine plant study. Yeah, so one idea is one idea shared here. She's here or not? She asked about if she wants to do the QTL. So what's how to choose the different metabolites? For example, QTL usually have a population, right? So those population, which way is much more convenient for you to quickly identify what is my interest one. As I before I said, if we have 100 the individual population, doesn't matter what's the back clone or the neo population, what kind of different genetic population, doesn't matter. But for metabolite, you want to screening all those, you want to check the difference. The quick method, you just use the very unique or identical condition, the metabolite, the peak, total iron peak. Use LCMS or HPLC, you can see the total metabolite shown up. Or use the different way, for example, extraction, sometimes you use methanol, sometimes you use chloroform or other different way. Anyway, you want to display as more as possible, display their difference, but also the consistency, because those have a similar genetic background. They should be have very stable, very good reproductivity in each individual, show very mostly similar, but still have some difference, different peak. You only focus on that peak. Then you see which individual have those peak, which don't. Then you corresponding to the molecular marker profiling, say SSR or other wrapped those metabolites and those marker finding. The high density marker is definitely the best, like a SNT. Those need to do the resequence or partial simply like sequence. So that you can find the SNP as more as possible. High density will help you define the single gene. So that way you match those metabolite to your marker, biomarker, or gene marker. If SNP, you will see whether some of SNP, you see one fragment of SNP always show together with one peak. As long as my SNP show in this plant, and they will show this kind of peak. That means co-segregation, right? The population, after the crossing, many genes will segregate. The segregation separates the marker and also separate the metabolite. Metabolite, the biosynthesis capability. So under identical condition, the growth condition is the same, and also other thing. So you will see the co-segregation of your metabolite. Then you just focus on identifying what that peak is. So that's what is great. And then also, if you already have some other target, then you just follow that. Then use the marker part. You need to do high density marker. Yeah, beside the SSR or others, you try to get the high density marker and fix and find the co-segregation biology marker as the metabolite. So that's the whole QTL works, right? Workflow just like that. this way. So we will continue. So the genome sequence is very important. Right now, like previous, we also show that the high-profile paper usually need to be assisted by the genome sequence to find some new genes. Then always use the transcriptome to quickly find the transcripts that could be associated or could be encoding the enzyme that catalyze the specific pathway. So that's very important. So the genome sequence can give you so many SNP marker. Why the bottom show the SNP marker very important. About the evolution story, that's probably you just know that the T have around 80 million years ago diverged. They have another WT, the common WT is the WGT, whole genome duplication. The first in million, 100 million, or 130 around. This is the gamma events, right? We know all plants have that. Then another whole genome duplication in 30 to 40 million, and CSA, assembly type, and CSS, synthesis type, diverged from the common ancestor at about 1.54 or even shorter years, but even closer. So the most important is they give SNP and SR density or TE those to help us future use the reference genome to do the major crop trait study to define the major crop trait. It determines by which genetic loci. That's very important. Yeah, doesn't matter you use the whole GWAS study, because like T plants very difficult to generate the population because there is the first crossing, then you get the population 1, and second time you cannot crossing because the outcross plant, the self incompatible. So that's why the generate population is very difficult. So GWAS, in that case, it become the popular. This is just for the evolution. This is for catechins. So first we do the genome sequence and we found that SCPL is very important for the gallulated catechin biosynthesis. Yeah, all those we do the tissue specific expression, the gene co-expression analysis with this, all those different type of catechin molecules to profile those. And this is also another called, we define the CSTS1, probably one key gene for thin biosynthesis. This is another paper to show that caffeine actually is also, they analyze a wide variety of tea plant and also non-tea plant, like camellia flowering plant. Camellia flower in one of the major ornamental plant, we just show their tea flower color, the very beautiful. So compared with all those plants, they find the caffeine only shown in C section, in C section. Those section plants actually currently, including some wild-type tea, they also can be used for making tea, just because they have caffeine. They have F-catechin and theine to make the tea functional to the human health. But one of the major findings find the camellia species compared with the cacao or caffeine, the plant, their unmethyl transferase, the gene family was expanded deeply in tea plant and also other the caffeine continuum plant. So we can put the NRT is very important. This is another genome sequence of different variety to show that there are many transposon insertion typical type or copy type, all those. Then those also can help us to understand atomic and the synthesis type of tea plant, their evolution. There are some also do the sequence larger amounts of local varieties of land species. Can do some GWAS analysis, at least predict some protein enzyme. It's very important from one of amber plant to the shrub, or from other. This generated a very specific type of metabolite in tea, either caffeine or catechin or theanine. So eventually will demonstrate how the genome variation causes the metabolite less diversity. Because in modern tea cultivar, usually the genetic diversity is decreased because of both the human selection or the environment selection. And this is for every tea. They find through the various the parameter of the population, genetic fluid between the population, because it's all the crossing plant. So they are very frequent. Also, this the LD distance shows that the landrace or wild type tea have different LDs. Actually, those they also you can track some relationship between the landrace and the Asian plant are also wild type to the modern tea cultivar or called elite. There's some relationship. And this is for QTL. For example, one of the major findings is those LOX gene or LAR, and also CBF. This is for cold tolerance. We know the tea plants originate from forest, the tropic forest. Our some is like a subtropic, but now the tea plant growth region can be extended to the temperate. Right? The summer already go to very low, into very north. So the increase the cold tolerance is one of the major change. So we can find those genes actually expressed and improved. The WRT or UTR, those, they also did the metabolite profiling in larger number of local varieties, find some. They also did the transcriptome to find the transcripts have a variation, have mutation. So this we can call this single nucleotide polymorphism. Those SMP even can guess from a transcriptome. So we even don't need to do resequence. We just use a transcriptome can do the QTL mapping. You use the transcriptome, gets the SMP, and those are also very effective. To high expression level, right? This is like the dominant phenotype. Yeah, transcriptome is almost like the dominant phenotype because they express, they show that. Not like genome sequence, some of they don't show any effect. So you use the transcriptome analysis, SMP, or expression level also can do the correlation with the metabolite to get the very good the correlation between the metabolite. In this case, it also did that. And also analyzed like around 200 the annotated metabolites, which is pretty confident. Those work actually is another university which we often communicate about the metabolites. They identify those peaks one by one. So it takes a very long time to, but still finally still have some clause. So, but anyway, those are some findings, just like the genome population structure analysis. They also did those metabolite visualization and show some. But their conclusion is very interesting. Each phylogenetic group contains signature, the metabolite. Means each, if it includes around 200 different varieties. Yeah, around 200 different varieties. If we do classify those according to metabolite profiling, we will show that each group have a different accumulation. That means the geo effect. Yeah. Geographic, the localization effect affect the plant metabolite profiling, and also those probably also because the plant is originated from different population. And in CSA, we think CSA is older, like CSS is more advanced because small leaves represent the plant from tropic area to expand to the northern or the colder areas. It's larger. Those CSS plants later on we also see have more fragrance. And also the CDF or ICE, those cold resistant expression related genes express the higher, and also from the genome level they seem to have some duplication, so expanded, so their cold tolerance increase. This is the metabolite can distinguish each group. This flannel, this new found flannel is very important. Like the routine or samanoside, the first glucoside or camphophyrol, those compounds actually very important. After that is the procyanidin, group like GC, ECGC dimer, or different dimer, trimer, those compounds are also phenolic compounds. Those show the different they think those should be a metabolite signature for different group of tea plant varieties. So is that
Yeah.
huh? Okay.
So rest of the things we will discuss in the next class.